Hi, I'm George from the Ensembl team. In this video, I'm going to demonstrate selecting genomes to work with in the new Ensembl site. Selecting genomes is the first task you will need to do before you can begin searching for genes and other features and viewing those in our other apps. To begin, we're going to use an app called the Genome Selector. You can get to this app by clicking on the link on the front-hand side page or on the top left corner. Here you will see a search box that asks you to find a genome. In this box you can enter into the common or scientific names of organisms across the tree of life. This site supports all the tree of life, not subdivided as our previous site, so you can search as you like to see if we have that genome available. I'm going to start with an example to search for human. If I click find, I will see the results. I have a count which displays how many genomes we have supported that match the search term. The genomes that are best supported and best evidenced rise to the top. So the top of this list contains the GRCH38 Human Reference Genome. There are two versions here. There is the integrated release version and a partial release version. Integrated releases are more stable and they link data across the site, whereas all data comes out as a partial release when it's newly added to the site. These may be more fresh, however, they are not as stable as they may be superseded if updates come before they are integrated. To begin working with the genome, I will select it and add it as a genome I'd like to use. It then appears as a lozenge on this top navigation bar. It gives me the name and the identity of the assembly that I selected to use. I can click on the lozenge, and within the Genomes Selector app, I will see assembly statistics and genome statistics based on the gene set and any other attached data, including variation and regulation data. To return to the search menu, I click back onto the Genome Selector app, and then I can begin searching other genomes I'd like to use. Say, for example, I'd like a plant genome, such as wheat. I type in wheat, I click search, and here are all the terms that matched wheat. I see bread wheat and durum wheat have risen to the top, and I can select both of those genomes to add up for me to explore in more detail, either in the Genome Selector app or by running searches through this app to other apps. I'm also able to search by groupings of genomes, so using taxonomic terms to group for a set of species or a set of individuals. For example, if I'm interested in fungal genomes, I can run the search for fungi, and I can see all the fungal species that we have available. We have over 388 right now. There will be more to come. Top of the list is baker's yeast. I will select that and add it up. I can also run queries for bigger selections of organisms across the tree of life. So I can choose, say, bacteria, run that, and then I'll have the list of bacteria that we have available. Popular users of Ensembl bacteria will know that there's many bacteria to come. We'll see those being added to the site very soon. I can select Escherichia coli and add it up as one of the species I'd like to work with. Here on this list, we can navigate between the genomes that we have selected. I can go back to see the human genome that I selected before. I'm also able to click and drag to move these around to see the genomes that I'm working with. If I happen to run a query for a genome, again, for example, human, and I see that one of the icons is grayed out, this is because I've already added up this genome to work with as a lozenge. Another example where if you choose the partial release for the same assembly base, you will get some extra metadata shown on the lozenge by view of a date. So this is the date for the partial release of the gene set attached to the human reference genome. And if we scroll back, we can see, there is the integrated release human genome there. When we start to add genomes as we work, we may find this list becoming a bit clunky and we want to remove some. There are two ways to remove. You can either click the little X icon on the left-hand side of the lozenge and remove that genome from our selection, or you can click onto the Manage Genomes icon, and then you will have a list of genomes that you can choose to either enable in apps using this toggle, or you can select genomes you no longer wish to use and remove the genomes from your list. When you're done, you can return to the genome selector and begin searching for other species and genomes that you want to work with. On the bottom of this page, we also include quick links to popular selections of species. So for humans, we have a nice link that leads us to a collection of human pangenomes that we are able to explore and use. These include loads of new data from the Human Pangenome Reference Consortium. If I navigate back, I can also click on this header, which leads me to external projects that are collaborating with Ensembl, including the Vertebrate Genomes Project, the Darwin Tree of Life Project, and I can use these links to find a quick link to all the genomes that we support that have been generated and produced by those projects. When I'm ready to begin searching for genes and features, it's helpful to return to the genome selector and be ready to use the second search bar on the list to search for a feature. But that we'll cover in another video. Thanks very much for listening, and happy genome selecting and browsing.